Welcome to another presentation on evolution from National Math and Science. Today we're going to be looking at macroevolution. This is part one of 4 parts on macroevolution. Specifically today we're looking at phylogenies. So you might be wondering, what do we mean by those terms? So let's start with macroevolution. Macroevolution refers to changes that result in new species. Most often that's what we mean by the terminology of macroevolution. It's also called speciation. But I'll warn you, you will find some authors in some textbooks you read that might use the term macroevolution to be talking about the development of complex structures like the eye. But we're going to use it today primarily to refer to speciation. And in this particular part, part one, we're going to be looking at phylogenies, which is a way to talk about the results of this process of macroevolution. So how can we, as biologists, represent the products of speciation? So we're looking, in other words, at evolutionary histories, at the family trees, sometimes called phylogenies. All right, so that's like the family tree for a bunch of species, showing how they're related to one another, and that's where we're going. So here we go. So first, if we're going to understand what we mean by speciation and relationships, we need to look at all of taxonomy, the way we organize life, in order to study it more easily. This field of taxonomy, or classification, was originated in its almost modern form way back in the 18th century by a scientist named Carolus Linnaeus. Linnaeus used the scheme you see on the left. With the exception of the very bottom, he did not have these domains down here. His broadest category was the kingdom, and the example we're looking at here is a panther. The panther belongs to the species Panthera pardus, which just means the specific type of cat that is this spotted panther you see in the picture. And more than one species falls into a broader category called a genus. Each of the genuses are grouped together into a larger category family, in turn into orders, classes, phyla, and finally the kingdom was the largest group that Linnaeus invented, the most inclusive group. In today's understanding of taxonomy, since the 1990s, in fact since 1990, we have had another category that's even broader than kingdom, and that's the category of domains. And we'll look a little bit briefly at why we need domains, why we needed a category that was broader than a kingdom. But keep in mind as we look at these, the goal of taxonomy is to represent the evolutionary relationships of all species. When Linnaeus started it, he was simply trying to categorize based on physical traits, physical characteristics, just so that it would make it easier for him and others to study life on Earth, see how it was organized. They weren't trying to determine evolutionary relationships because he had no idea about life literally being related, one species to another. Today we have a much better understanding, and we still look at his system, and it's amazing that in many cases the traits that he chose also end up being indicative of actual evolutionary relationships. By the way, Linnaeus only had 2 kingdoms. He looked at things as either being plants or animals. That was as complex as he thought they were. If it was green, it was a plant. If it moved around, it was an animal. Since then, biologists have added some other categories. In the 1800s, starting in about 1866, due to the invention of microscopy, scientists started discovering creatures that weren't clearly either plants or animals, little things that could swim around but were green like plants. And they invented a kingdom called Protista. And then later, in the 1930s, they added a kingdom called Monera for those microscopic organisms without nuclei, like bacteria. And then finally, in the 60s, we added a category for things that didn't move. They looked sort of like plants, but they were not photosynthetic, and those are the fungi. But starting in the 70s and 80s, the Monerans, the kingdom that had all the prokaryotes, were split into 2 separate groups, 2 separate types of prokaryotes, and we'll look at those in just a minute. And here we are. So, fairly recent, the invention of the term domains. In fact, it was established in 1990 and became accepted pretty quickly. due to the work of a scientist named Carl Woese. He's one of my favorite scientists. He did some amazing genetic research, and he discovered that if you looked at a piece of genetic information that is conserved, meaning it's found in all life on Earth, he could determine relationships based on that genetic information. And what he looked at was ribosomal RNA. It turns out that every life form on Earth uses ribosomes to synthesize its proteins. Within those ribosomes are structures of ribosomal RNA that are critical to the functioning of those organelles, ribosomes. You might think that since ribosomes seem to work in a very similar manner, the rRNA, ribosomal RNA, would be very similar. And it's similar to a point, but Carl Woese was able to find distinct differences among different types of life. But he also found commonalities among what we thought were very different groups. So for instance, all of the eukaryotes, plants, animals, fungi, protists, all of them have ribosomal RNA that is more similar to each other than it is to the ribosomal RNA of the prokaryotes. So he determined he needed a category that would lump all of those four kingdoms together into a broader classification. And that's why he invented the term domain. And you'll see in the little tree on the left, eukarya is the name for all of what we used to consider four separate kingdoms of eukaryotes. So they still have the kingdoms, but those kingdoms are underneath a broader category called the domain eukarya. But then what was almost equally striking to him was to discover that there were distinct differences between the two groups of monerans, the two groups of prokaryotes. Turns out that what we think of as modern bacteria, like E. coli that you have in your gut, are very different from some other prokaryotes that he named the archaea, meaning the ancient ones. And these were prokaryotes that lived in very extreme environments. They could live in temperatures above boiling. They could live in acidic environments. They could live in environments that were so salty nothing else could survive there. So these bacteria, when he sequenced their ribosomal RNA, it turns out that the bacteria had significant differences from the archaea. Different enough that he thought they deserved their own domains. So we now have three domains, eukarya that we mentioned already with all the eukaryotes, but what's really important to understand is that the prokaryotes are not all that similar to one another when we look at their rRNA, so we give them two separate categories, the archaea and the bacteria. And what was a third astounding discovery was that archaea actually had more in common in the ribosomal RNA with eukaryotes than it did with bacteria. So we see what's called a cladogram here on the left, a branching diagram that shows those three domains. And you might wonder what was present here at the beginning. What was the ancestor of all life on Earth? Many scientists think it was probably archaea due to the extreme environments they can survive in, but we don't know for sure. But we could have had archaea here at the beginning, and then there was a branching event, meaning the archaea split into multiple populations, and in one population developed this line, which in modern terms are bacteria. Another group of archaea branched off here, or this is the original group actually after the bacteria branched off, and then we have archaea that branched again into the eukarya and then continue to have archaea all along. So that's one way to look at that cladogram. So a couple terminology terms here for you to know. You should know what a taxon is. If I refer to a taxon, we simply mean any level of hierarchy. So a domain is a taxon, a kingdom, a phylum, and so on. So let's look at phylogenetic trees. Phylogenetic trees, and the term phylogenetic tree and cladogram are often used interchangeably by biologists, and I think you'd be just fine on an AP biology test using those terms to mean the same thing. But I'll give you a little technical difference in case you're interested. A phylogenetic tree is a representation of the actual evolutionary history of a species. Okay? The term phylogenetic tree we think is used when we think we have the actual true evolutionary relationships represented in the tree. And in those sorts of trees, the length of the branches are significant. the ordering of the branches can be significant. A cladogram, on the other hand, is a hypothesis about relationships, but may not be based on the full picture. So a cladogram could become a phylogenetic tree, but we'll look at some differences as we go along. But for our purposes, we're going to assume in most cases phylogenetic trees and cladograms refer to exactly the same thing. The term you do need to know here is phylogeny, which just means studying the evolutionary relationships, like the family tree of species. So on the left we have an example of 3 different cladograms. These would be cladograms showing you hypotheses about the relationships between these 3 groups of birds. So how do we draw them? How do we determine which species is more closely related to which? We use evidence like morphology, which just means structure. We use the fossil record to see which species arose first. We use embryology, studying how the embryos develop. But the best evidence by far that we can use is in the molecules of life itself. The very best, the supreme evidence that we can use to determine relationships would be DNA, when it's available. Obviously with fossils you don't always have DNA. Sometimes we do, sometimes we don't. But DNA is the best evidence for relationships because the only way to get DNA is from your parents, who got it from their parents, who got it from their parents. So if 2 species have a lot of DNA in common, we assume they must have had ancestors in common because that's the only way they could have acquired that similar DNA. Okay, so DNA is the best evidence we have. So here's just some terminology for you. I want you to pause it here and take some notes, read the whole slide, and that's what you do. All right, you back with me? Notice we have those definitions I already gave you of cladogram and phylogenetic tree. So here's a specific phylogenetic tree that shows the relationships between Sirenia, which are creatures like manatees over here on the left, and Proboscidea, fancy word for big nose, which refers to the elephants. So in this phylogenetic tree, we have a representation of what we believe to be the true evolutionary history of elephants. Notice many of the lines don't make it all the way to the top, like this one, and this one, and this one. All of those branches that don't make it to the top line represent species that are now extinct. So we now have 3 groups of elephants that are extant, alive today. None of the other elephants in the picture are still around today. They're all extinct. And then we have Sirenia over on the left. So what this phylogenetic tree is telling us is that manatees are closely related to elephants. And there's lots of evidence that supports this. We are confident that manatees are more closely related to elephants than they are to walruses and seals. And just some basic biology about them will help reinforce that idea for you. If you think about what elephants eat, they are herbivores. They only eat plants. The same is true for manatees. They are also herbivores. And if we look at their biology, their DNA, their molecular structures, they have much more in common with elephants than they do with things that on a superficial level look sort of similar, like say a walrus. Walruses and seals are carnivores, and they are not closely related to the manatees. So manatees have a more recent ancestor right here in common with elephants than whatever ancestor they would share with other animals, including walruses. So what do we really mean when we say that manatees and elephants are related to each other? Do we mean that elephants descended from manatees, or vice versa, that manatees descended from elephants? What do you think? Hopefully you would say no to the second question. Just being related to does not mean the same thing as descended from. I can give you a real quick example to illustrate that. Think about your own family. I'll use my family as an example. I've got a brother, I've got cousins, I've got friends. And if we can consider what these, we'll say we have 3 men that we're comparing to me, my brother, my cousin, and a friend of mine. If you could look at them, if you could see me standing there and see these 4 men, the 4 of us all standing together, and I asked you to pick out which one was my brother, I'm willing to bet you could determine which was my brother just by looking at us. And why is that? Obviously, my brother and I look a lot more alike than I look like any other men on the planet. I look much more like my brother than I do the other gentleman. So the next question is, why is that? Why do I look more like my brother than I do anybody else? You can tell me. Hopefully you have it figured out, because I have the same parents. So we could draw a little evolutionary diagram that shows me and my brother, and we have my parents, my dad and my mom, as our direct ancestors. So we have common ancestors, my parents, and the reason we look so much more alike than we do like anybody else, because we inherited DNA from our parents that is identical. My brother got copies of some of the exact same DNA that I got copies of from my parents. So most of our DNA is exactly the same. It's not just similar, it is exactly the same. Now what about my cousin and just a random friend of mine? Could you determine by looking at them which one was my cousin? What do you think? Think about your own cousins if you have some. In my particular case, my cousin looks a lot more like me than any of my friends do. And why should that be? Well, same idea. I'll put my cousin here. Turns out that my cousin's parents include my aunt, who is my mother's sister. So my aunt and my mom have the same parents who are my grandparents. So my grandparents passed some DNA on to their children, which are my mom and my aunt. So my aunt has some of the exact same DNA that my mom does. When she gave birth to my cousin, she passed on some of the exact copies of some of the exact same DNA she got from her grandparents that my mom got from my grandparents and passed on to me. So my cousin has a lot of the exact same DNA that I do, and as a result we look more similar to one another than I do to somebody who is not so closely related. So we can say that my cousin and I are related, and when we say related, it means we share common ancestors, in this case my grandparents. We could also use a phrase like more closely related to. I would say that my brother is more closely related to me than my cousin. What does that phrase mean to you, more closely related? Looking at this tree, hopefully you recognize right away that my brother and I have more recent ancestors in common than my cousin and I. So having more recent ancestors and therefore more DNA in common means more closely related. So the key marker is the amount of DNA that we have in common. My brother and I have much more DNA in common than I do with anybody else on the planet. My cousin and I have more DNA in common than I do with anybody who doesn't share my same grandparents. What about my friend over here? It's a friend of mine named Doug. Alright, so Doug is over here. Do you think he and I have anything in common? It would be the same thing as asking, do you think you and I have anything in common? And the answer is, of course we do. All those things that make us human, we share. And we share the genes for all of those structures as well. I'd bet that you and I have virtually identical genes for most of our proteins. Our hemoglobin, most of the proteins that make our bodies run, yours and mine are exactly the same. So are mine and Doug's. So we have many traits in common. That means somewhere, I don't know how many generations back we gotta go, but somewhere back here I can guarantee you that my friend and I have an ancestor in common. How do I know that with such certainty? Because my friend and I have some of the exact same DNA. And the only way to get DNA is to get it copied from your ancestors. So all of us that have DNA in common must have ancestors in common. So am I related to my friend Doug? Of course I am. I'm related to him because we share an ancestor. I know that because we share DNA. I'm more closely related to my cousin than my friend. I'm also more closely related to my brother than I am to my cousin. Starting to get the picture? Hopefully. So now, what about this question of descended from? Would it make any sense at all to ask if I'm descended from my brother? It makes no sense. How about my cousin? No, I'm not descended from my cousin either, even though I'm related to him. And I'm certainly not descended from my friend Doug, even though I am related to him as well. So hopefully that little illustration makes it clear to you there's a big difference between being related to a species and being descended from it. So if we back up for just a minute and look at the manatees and the elephants, modern manatees and modern elephants are related to one another because they share an ancestor in common down here where I'll put this big X. That ancestor at that branch point is shared by both manatees and elephants. It gave DNA to all of its descendants. So manatees and elephants have a fair amount of DNA in common. But are either one just descended from the other? No, it makes no sense at all. They are related to each other, but not descended from each other. Okay, so let's move on and keep looking at how we can represent these relationships. So what we're really representing is the result of macroevolution, the result of speciation. So here's a phylogenetic tree based on Linnaean classification, which is just looking at structures. In many cases, just looking at structures does provide what ends up being an accurate evolutionary history, but we don't know it for sure until we can compare DNA and other chemicals of life with one another. Okay, so sometimes, like it says here, advances with DNA and protein may result in changes in the traditional classification. So I ask you again to think about why is it that DNA is better evidence for evolutionary history than simply looking at physical traits. So let's get a little terminology straight here as we look at branching diagrams. This is a cladogram. And when I say a cladogram, what I mean by that is that clade means branch. In this case, the length of the branches is not indicative of time. We're just trying to draw out relationships here. So we've already talked about the term taxon. Plural for that is taxa. So I can see multiple taxa here. 6 different ones, A through F. Another term to know is node, which is just a fancy word for a branch point. So how do branch points arise? Imagine I have an ancestral population of some organisms, all the same species living in where that circle represents. And they split into 2 separate groups, maybe migrate to different islands or different parts of a territory, and the natural selection treats them differently in those 2 environments. If you're with me for the study of microevolution, we talked about this process a little bit. So once they split and then accumulate differences over time, we might end up with entirely different species represented by these taxa. Another term, sister taxa. Sister taxa simply refers to species or categories that share a common ancestor, a recent common ancestor. Okay, so B and C there are sister taxa. We would not say that A and D are sister taxa, even though they do share a common ancestor way back here. There are ancestors in between that they do not share. Okay, another slide giving you that same sort of terminology. We have a new term showing up here that I haven't talked about yet. Polytomy or polytomy. Polytomy. What does poly mean? It means many, and we're talking about many branches here. So what this represents in this cladogram is that we can't tell which diverged first, D, E, or F. The degrees of relationships and commonalities between them, we know they're related to one another, we know they share a common ancestor, but we don't know the order in which they diverged from one another. But we are confident that in every case, species diverge into, when they do diverge, they diverge into 2 species at a time, not 3 at a time. So the hypothesis is when we see one we can't determine exact branching orders, we call it a polytomy, and it just means we don't have enough evidence yet to tease out how that branch worked. So if we look at the top case, here we have 3 taxa that are all related to a common ancestor at a branch point very similar to the one in the polytomy, but now we have enough evidence to know the order in which they branched. So we had a common ancestor, taxa A branched off into this common ancestor here, where the X is, which in turn branched into what I'll call Y and Z, taxa B and C. So the hope would be that eventually we could resolve the questions about the polytomy and get a cladogram similar to the one for the first three. Take a minute. I want you to pause the slide here and look at the two drawings on the left and the right and see if you can determine how they're different or whether they're the same. Okay, so hopefully you did that. I'll remind you, if you're new to this, you should be pausing the presentation whenever you need to take notes. Rewind it and listen to what I've said again. I know I talk fast sometimes, but you can pause this at any point and work through it at your own pace. So two different phylogenetic trees here illustrating the same relationships. And my question was, are they the same? Are they really the same? And in fact they are, even though visually they look a little bit different. You can rotate any of the branches. So this first branch point, we separated A, B, and C from D, E, and F. We did the same thing on the right, A, B, and C from D, E, and F. And it doesn't matter that we put A, B, and C on the bottom on the right one and the top on the left one. That has no significance. They represent the same relationships. Now notice we can also rearrange within here. It doesn't matter. I could change this B and this C and switch them and make that B and that C, and it represents the exact same relationship. So rotating branches has no effect on the accuracy of the drawing. So now let's talk about cladistics and the terminology there. Clade means a branch. So a clade, as is defined here for you, is any taxon that consists of all of the evolutionary descendants of a common ancestor. So look at the different colors. In the yellow rectangle, that's a clade, but it's within another clade, which is a blue rectangle. The sort of salmon colored on the right is a clade, but the entire diagram could be seen as a single clade as well. Now, while we're here, let's take a look at how relationships also result in taxonomic categories that you're familiar with. So each of these branch points, each of these species, let's call them A, B, C, D, and E. So the tips of the branches represent species. What would you call a category that has two species in it, like D and E? So we could consider this salmon colored. That could be a genus that contains species D and E. A and B might also be a different genus. Now what we call categories that have more than one genus within it, we could look at this entire grouping, and the category that's higher than a genus is a family, so that could be a family in taxonomy. And a group that has a broader grouping than family is order, so the entire picture could be the order that contains all of these species. Okay, hopefully that makes sense. So you know kingdom, phylum, class, order, family, genus, species. So their groupings reflect their evolutionary relationships. Here's some simple terminology for you. You should just pause the slide here and jot down these. I'm not going to read them all to you, but they're all important terms. Okay, let's move on. A couple more terms that describe the processes of evolution of producing new species: anagenesis and cladogenesis. Clade again means branch. I'm just going to make that one easy. Anagenesis is straight line evolution or phyletic change. So in the left-hand picture there we have one species. The entire population has evolved into a new species. Okay, that's anagenesis. Cladogenesis is defined here. Cladogenesis, we have the same ancestral species and it's branched into two species. In this case, the original species is still present, and now we have a species descended from it. That's the second species. Now we could have had anagenesis along this line right here, so we could have had this evolve into even a different species, or these could branch again and again and again. Okay, so cladogenesis, branching, usually involves physical separation into separate groups. in the evolutionary history. And then they were treated differently by natural selection, and different species resulted. So what do you think's been more common in the history of life on Earth? Anagenesis or cladogenesis? Do you have an answer? I can't quite hear you, but I'm going to assume you said cladogenesis. And the reason you know that is because the number of species has increased over time. We can tell by looking at fossil record. The only way to increase the number of species is by budding or branching, cladogenesis. Anagenesis doesn't increase the number of species. It changes the species, but the number would stay the same. Okay, now both have occurred in the history of life, but cladogenesis is probably much more common than anagenesis. So a little bit on how we create these phylogenies, these branching trees. So here's a little bit on the fossil record. Did you get it all down? It's pretty common sense here. We're just saying that older fossils are found in deeper rocks, deeper layers than younger fossils. Now, some interesting terminology. Homologous here refers to features that are shared because they came from the same ancestor. So my brother and I have many, many, many homologous features. In fact, virtually everything that we each have was inherited from our parents. So we got them from a common ancestor. So the structures my brother and I share, the molecules we share, are homologous. Now, what if I had a mutation arise in me that my brother didn't have? That would not be a homologous feature. Right? That would be what's called a derived feature, something that arose after inheritance from a common ancestor and didn't actually come from the ancestor. Okay, so we're going to look at that over and over again here. Homologous features turn out to be really, really important in creating evolutionary trees. I like the art on the left. Show you again difference between anagenesis and cladogenesis and how both could occur even in the same evolutionary line. So here we have anagenesis. Let's say we have an ancestral species called A, and it's evolved along the way here, slowly accumulating changes until we have to give it a new name. We'll call it B. And then B could diverge. This might split into B, and then over here now we have C, which in turn split into D and E. So we could have multiple species arising, and along the way, both processes, straight line evolution and cladogenesis. Okay, last note here on homologous features on this slide. This third bullet is probably the most important one on the entire slide. Hint, hint, hint. Homologous features can be structures, like bones in a limb that we'll look at in a minute, or DNA sequences, which are the best features to look at in terms of determining homology and therefore relationships. Ancestral versus derived traits. It's like saying homologous traits versus those that arose not from a common ancestor. So some great examples here. The limbs on the left are homologous structures because they have very similar bones, and they inherited this pattern of bones, one upper arm bone, two lower arm bones, a group of bones in the wrist, and then a group of bones that are like fingers. That pattern shows up in all of these individuals, even though they're very different structures. An arm, a leg of a dog, the wing of a bird, the flipper of a whale are all homologous because of their internal structure. And they reveal that they have a common ancestor. What about structures that look similar but are not really from a common ancestor? A great example would be the wing of a bird and the wing of a bat. So on the top here we have a bat wing, and on the bottom we have a bird wing. The bones inside them are homologous. One upper arm bone, two middle bones, wrist bones, finger bones. Very similar structures in both the bird and the bat. So the bones are homologous, but the wing itself is not. Birds and bats did not inherit wings from the same ancestor. Their wings evolved independently, and if you look at them, their wings are very, very different. Bird has feathers. Feathers are attached in a different way than the wings of a bat. Bats don't have feathers at all. They have skin that is stretched across these bones to make flight structure, but it's very different from the feathered wing of a bird. So those structures, if they are similar but not from a common ancestor, we say they are analogous. Similar, but not because they came from a common ancestor. Okay, so it's going to be really important for you to remember the difference between analogous structures and homologous structures. So how do you think that happens? How did it work out that birds and bats have these analogous structures, structures that are similar, without inheriting the DNA to control them from the same ancestor? How did it come about that they should have such similar structures? Got an answer? The answer is right here on the left. Similar selection pressures. It was advantageous in light of the selection pressure for flight, for the development of very similar structures between bats and birds. Both of them benefited from that structure, but they did not evolve them from the same ancestor. They did evolve them in similar selection pressures, in this case the physics of flight. So we have a term for that. Structures that evolve not in homologous terms, but in analogous ways due to similar selection pressures. We call that process, and you should write this down, convergent evolution. Convergent evolution. They converged on a similar idea, you might say, similar adaptation, because of similar selection pressures, not because the structure was inherited from an ancestor. Now, don't get me wrong, birds and bats certainly have an ancestor in common, but the ancestor that they have in common did not have wings. So it's represented on the cladogram on the right. So here's the ancestor that they all have in common. That ancestor had 4 F-O-U-R, 4 limbs, 1, 2, 3, 4. And the front legs in some lines of descent from this common ancestor, called 4 limbs, F-O-R-E, front legs, evolved into wings in bats and birds. But notice that they're not found in mice or crocodiles. So that tells you it wasn't present in the ancestor down here. It evolved independently. Okay, that's analogous versus homologous. Another example, I'll let you pause it and write this down. I'm not going to read it to you. All right, back with me. Another example of analogous structures. Okay, so I'm not going to read the whole thing to you, but a term I want to point out to you that's important here is tetrapod. What does tetrapod mean? Tetra means 4, pod means foot. So 4-footed, really 4-legged creatures or 4-limbed creatures, F-O-U-R. And that's all of the creatures that have 4 limbs, or at least remnants of 4 limbs. Even us humans are tetrapods. We walk around on 2 feet, but your arms are also limbs, so we have 4 limbs. We are tetrapods. So this is a neat story here about how whales and dolphins actually have an ancestor that lived on land, but they have structures, overall shape that's very similar to creatures that live in the water like sharks, even though they don't have a recent common ancestor. So the design, the structure of a flipper or a fin is very similar between mammals that live in the water and fish that live in the water because of the similar selection pressure in the aquatic environment. So the flippers of a whale are analogous to the flippers or fins of a fish, like a shark. Some other examples of homology versus analogy. First top half of the picture at the top, those are homologous. The wings in the bottom are analogous. Now, if you were with me for Macroevolution Part 1, we did a discussion of molecular clocks. Here's a review of that, applying it again to evolution and looking at evidence for creating phylogenetic trees. So we've covered this together once before. I'm not going to read all of this to you, but it's great information. You should pause and take notes on these next few slides, but I don't want to insult you by simply reading it to you. But the information is really cool, so go for it. Okay, get all this? More a little bit here about using the rate of amino acid replacement as a molecular clock. Again, we're talking about changes occurring at a steady rate so that we can calculate how long a structure has been evolving. Another great illustration. I love this one. It's a recent example of using molecular clocks to look at evolution within our own lifetimes. We can calculate how long ago HIV started diverging into the many forms around today by using a molecular clock to study the evolution of that virus. And this is a wonderful image that ties all of the molecular clock data of the last several slides together. Way too much for me to read to you, so you need to pause this and take notes on this cool information. All right, did you get it? Now I want to give you a little tip here. If you want to read more about molecular clocks, more about evolution in general, this image comes from one of my very favorite websites. And if you simply Google the phrase understanding evolution, okay, make note of that, and then when you get time, Google it, understanding evolution. If you Google that, the very first hit's going to be a website called Understanding Evolution from Berkeley University. Go to that website, and there's lots of information about evolution there that's really well presented. If you have time, work through their little mini course on evolution. It's called Evolution 101. And you'll know way more about evolution than we've been able to cover in these little PowerPoints together. Okay, so understanding evolution from Berkeley. So let's talk about how we can actually draw these cladograms, these phylogenies we've been discussing in this whole presentation today. A couple rules you need to know. When we look at data, we sometimes have data that could be represented in more than one way. We could draw multiple versions of a branching diagram that would represent the history we're seeing. So how do we know which one is correct? We use these two rules, maximum likelihood and maximum parsimony. So we have definitions for you here, terms and rules you need to know. Now let's look at some examples. So we have a data table at the top. Now it would not surprise me at all to see a data table like this on the AP Biology exam with them asking you to create a cladogram that represents the data in the table. I can almost guarantee that will occur. They'll do one of two things on the test. They'll either ask you to draw a cladogram based on a data table or create a data table based on a cladogram. They might even do both. So you need to be very comfortable with interpreting data tables and drawing cladograms. So in the example here, you see that there are two ways to represent the same data above. So which one do you think is more likely to be correct? Okay, so the diagram showed you the first one it says it's more likely. This slide explains why it's more likely. Okay, here's a sample that could be a question on the AP Biology test. Look at the data table on the left, and then once you've looked at it, pause the slide, and using that data table, I want you to do this first bullet. I want you to draw a cladogram that accurately represents the data seen. By the way, when you see this table, a 1 means that it is present, just like a checklist. So leopards have hair. Leopards also have an amniotic egg, and so on down the line. Okay, tuna have a vertebral column and hen's jaws. They don't have the others. So 1 is like a check mark. The circle or 0 means it's not present. Okay, see if you can draw a cladogram. Okay, so you're back with me. Let's look at what the answer is. So there is the correct version of this cladogram. Did yours look like that? Now you could have drawn it branching up. Could have drawn it branching down, orientation, top to bottom, left to right, bottom to top. All of those are equally valid. But the important thing to know is the order of the branches and where the traits appear. All right, so hopefully you did one similar to this. What you should notice is things like when vertebral column appears here, everything that branches from this common ancestor that's circled in red would have a vertebral column. So lampreys, tunas, salamanders, turtles, leopards all have that trait because they share that ancestor. The same way that amniotic eggs are shared by turtles and leopards and none of the other creatures. So amniotic egg here passed on to anything on that clade, but not the rest of the clade. So we can use the term clade several ways. This whole diagram could be a single clade. We could talk about the clade for the turtles and the leopards. Or the lampreys, tuna, salamander, turtle, leopard could be a clade. Okay, any group that includes the common ancestor. So let's see if you're really getting this. So I've got a few questions for you. I want you to think about each one. See if you come up with the answer before listening to my answer. Okay, so my first question is, what do you think is more closely related to the lancelet? The lamprey, the tuna, the salamander, the turtle, or the leopard? Are any of those more closely related to the lancelet than any of the others? What'd you say? Hopefully you said they are all equally related. So even though we have an order from top to bottom here, lancelet to lamprey, that order does not indicate degrees of relationships. So what do I mean by that? Lampreys and leopards are just the same degree of relationship with a lancelet as a tuna or a salamander. How do I know that? Because lampreys, tuna, salamanders, turtles, leopards, all of these guys have a single ancestor in common after they split from the lancelets. So where the vertebral column is circled, that represents the ancestral group that all of these guys share. So the differences between them and lancelets were established at this branch point right here. When that group diverged, the degrees of relationship were established. Okay, how about another example? What's more closely related to the turtle, the salamander or the leopard? More closely related to the turtle. Okay, hopefully you said leopard, because leopards and turtles share a more recent ancestor than turtles and salamanders. Okay, let's try one more. Is the tuna more closely related to the salamander or the leopard? Tuna, more closely related to the salamander or the leopard? Hopefully you said they are equally related, because the divergence between tuna and salamanders and leopards happened right here. So the divergence happened once, and salamanders, turtles, and leopards are all in the same clade, which is equally related. Everything in that clade is equally related to the tuna because they share one ancestor that is unique from the tuna. All right, that's it. Next, we're going to look at how speciation occurs. So in this part one, we were looking at ways to represent speciation. In the next part, we're going to look at how the act of speciation itself, how it occurs. That's all we've got for this one. I'll see you next time.